The overall goal of these procedures is to study how changes in gene expression, either within developing cells or in their surrounding environment, influence temporal processes during development.
This method can help answer key questions, such as whether the timing of fate specification is controlled by genes expressed cell autonomously within a cell or cell non-autonomously in neighboring cells. The main advantage of this technique lies in the ability to follow individual progenitor cells in an in vivo vertebrate as these cells are developing in genetically distinct environments. Demonstrating the procedure will be Stefanie Dudczig, a research assistant from my laboratory.
After preparing zebrafish embryos as well as microinjection needles with morpholinos or mRNA according to the text protocol, immerse the needle tip in a drop of mineral oil in a dish. Measure the injection volume by pressing the foot pedal and adjust the gas pressure and/or duration time until the injection volume is equivalent to 1 nanoliter as measured with the micrometer slide or alternatively with the eyepiece graticule. Place a microscope slide in a petri dish and with a transfer pipette, deposit single-cell stage embryos along the slide edge. Next, with a fine transfer pipette, remove as much liquid as possible to prevent movements during injections. Then, using a microload or pipette tip with half of the thin tip cut off, align the embryos into a single